When we study immune responses, we tend to always just get blood samples and look at cells in the blood samples and changes associated either with induction of inflammation or antiviral response or adaptive response, a specific response. But the blood, as far as cellular immunity is concerned, is only a vehicle to get to the actual tissue where the responses, the effector responses take place. And so we have kind of avoided looking at cellular responses, which are a really, really big part of the immune system. And again, whether it's innate or adaptive, we really need to look at how cells interact and how they respond to pathogens that are introduced in tissues, because that's where the pathogen is usually located. And it is actually quite a complicated situation that T cells are the major cell of cell-mediated immunity in vaccine development, and yet we only tend to look at antibodies because it's so much easier. And T cells are very restricted in what they can do. They are not that easily activated because they are the orchestrator of the immune response. And they are very restricted in recognizing their antigen in association with an antigen-presenting cell. And so right there you have that cellular interaction that's needed first to activate these cells, and then when you actually look at the effector response, T cells work in the tissues where the infected cells are. So inflammation, which is not T cell driven, occurs in tissues, and effector responses like cell mediated responses, T cell mediated responses occur in tissues, and yet we have very little knowledge at this point to actually evaluate those responses and have a close look at them. So what's new with AvID is that we have for the first time a minimally invasive window into cell-mediated responses, whether they're innate or adaptive, whereby we can actually look at temporal activities in terms of, with respect to time, and quantify the response, and then also looked at the quality of the response, all in the same individual. And we can also do this along with the blood, so we have a two-window system to actually look at these responses. The first thing you have to do is you have cells in the tissue that sense the infection. Then they send out signals to recruit cells from the blood that can help with this infection. That recruitment itself is requiring vascular changes that allow the leukocytes to leave the blood, to come into the tissues, guide them to the actual site of infection. And so there's a lot of communication going on. And then they also need to be activated in those tissues. So we're missing, if we're just looking at the leukocyte changes in the blood, we're missing all that interaction that is so crucial in actually getting immune response to occur, an effector response, and result in the elimination of the pathogen or the infection. Okay, the crowing feather is a skin derivative, and it has a piece of skin called a pulp inside the feather sheath, and that piece of skin consists primarily of dermis, and then that is enveloped by epidermis. So it's like a little column of skin that if you pull up your skin like this, that is available for study. So all these cellular responses occur in a complex tissue, and the dermis particularly has blood supply, so the cells can be called to that site if we introduce a test material. There's nervous tissue in there, there's fibroblasts, and there are sensing cells like macrophages and dendritic cells that can respond like they would in a skin. And there I really want to bring people back to the human medicine, like of allergy testing, where you basically make a grid on the back of on the skin of your bag, and then put the different allergens into each square, and then look at the allergic response separately. And that's very much like what's going on with the crowing feather. I think it's even more contained in that I consider it an in vivo test tube just from the shape it has, that when we place one test material in one feather, you get that unique response in that feather. The other feathers are not affected by that, and if you really think about how immune responses are, local immune activity is initiated, it always involves changes in that tissue, sensing in that tissue, and then just changing the vasculature in that tissue for the cells to actually come in. So yes, we know we can do different dosages of the same test material in the same animal, as long as we map out what we injected at what site. And then we can also do different test materials. And at this point, I've done 4 different test materials in the same animal and got very unique responses. And then also the dosage. We have an experiment going on right now where we would put 3 different dosages of the same material and the vehicle response in the same bird in different feathers, and as long as we know which feather we injected with what, we can definitely keep track of that. In vaccine development, the use of AVID varies depending on the questions you want to answer. If you want to answer what is happening when you introduce your test material, what does your adjuvant contribute to the immune response, what is happening when you first introduce the vaccine into a complex tissue, then we would directly inject the adjuvant alone or the individual components of the vaccine into the growing feather, and then we could collect for the feathers at the various time points, like within minutes or hours after the injection, to look at the really early events, and then carry it out maybe through 7 days, and get sort of from a first introduction of the antigen to a resolution of that immune response. Now, if you want to look at a recall response, meaning that you vaccinate the bird, say intramuscularly or subcutaneously, whichever route is used for your vaccine, and you then let that bird go through an immune response. And what we typically do is also take blood samples and look at the antibody response for, say, 4 weeks. And then 4 weeks later, when the memory response would have developed, you can inject a crowing feather and then look at the quality of the cellular response, or what is actually happening at the site where the antigen is encountered. There are also different ways on how you can also look at the effector response. If you just step back and think about the antibody response, so we vaccinate an animal, and then we take blood samples to look at antibodies, say 5 days after the vaccination, 7 days, 10 and 14 days, and we see that rise in antibody levels, and that is basically the effector response that has been initiated in the secondary lymphoid tissues. So for cell-mediated responses, then what we have done, we have injected the crowing feather with the antigen, say at the height of the antibody response, knowing that this is now the first phase of the actual effector response, and then looked at the effector cells being recruited. So this is not a memory response, but this is an active primary response where we have opportunity now to actually look how quickly do these effector cells come in, and what are they doing in the tissue. Some of the vaccinations go out into adulthood of the birds. Like if you want to look at a memory response in an adult chicken, from from the experimental side, how do I say that? That question just has come up in the adult, because birds, when they go into production, they actually have their mature plumage, and so they don't tend to have crowing feathers. Now, to produce crowing feathers, we usually pre-pluck before an experiment. We pull crowing feathers, so they have to have some living tissue attached with them. They come out easily, and we make sure we have a row of crowing feathers, and they regenerate, and 18, 19 days later, they're just perfect individual little test tubes to use for the study. Now, I've also done this with birds that past 20 weeks of age that are in maturity, and what the approach there is to actually be sure to pre-pluck when they still have crowing feathers, and even if 3 weeks later you don't use those crowing feathers, pluck them again, they will regenerate those, and so we can go actually quite far into adulthood. So what are the endpoint measurements, or what do we actually do with in the laboratory with these biopsies, basically, that we take at different times during the response that was initiated by injecting the test material into the feather. So we basically pull the feather, the crowing feather, and what's really nice about taking a biopsy like this is the crowing feather is not anchored into the tissue like a mature feather is. And so they come out very easily. There's no obvious pain associated with doing that for the birds. And then we take that feather, and we have now quite a big piece of tissue. Like I said, it's about 10 millimeter by 2 to 3 millimeter piece of tissue. And then we can make single cell suspensions, and then label, do immunofluorescence staining, where we then can very specifically identify the cell types that have been recruited in response to injection of the test material. And we can do cell population analysis by flow cytometry, which is very same day, very fast results. We have also done a lot of using that tissue to isolate RNA, so we can look at cytokine expression, actual functional activities of these cells through that process. Unfortunately for the Avian system, we don't have that many assay systems available yet to look at the actual cytokine proteins, but that is something we're also working on. We can do frozen sections and then actually localize the cells that have come into the tissue, identify them by immunohistochemistry, and there we can actually see if there's obvious antigen present, like viral antigen or bacterial, which cell has phagocytosed, or what is the general activity and interaction going on, for example, with heterophils coming in, and then they are dying, and then the macrophages clean that up, or the heterophils can have this net formation. So all that can be very easily visualized through histology. And again, we have quite a big piece of tissue at each time point, and we can then really establish what an individual actually does in response to the injection of the test material.